close your eyes. Imagine a single human cell, something so small that millions of them could fit on the tip of a needle. Yet, inside each of those tiny cells lives something even smaller, something more powerful than it looks. A delicate spiral, gently coiled like a staircase made of light. This is DNA, the blueprint of life, a soft thread made of instructions, quietly working behind the scenes, telling your cells how to grow, how to heal, and even what color your eyes will be. The full name of DNA is deoxyribonucleic acid, but you don't need to remember that. What matters is this: DNA is the instruction manual that makes you, you. Let's begin at the beginning. Every living thing, from a sunflower to a blue whale, carries DNA. It's the universal code of life. But instead of being written in letters like A or B, it's written in chemicals. And even though this code is very old, it still works beautifully, like a poem passed down through generations. So where is DNA found? DNA lives inside almost every cell in your body. Most of it is tucked safely inside a part of the cell called the nucleus, a small round space near the center. Think of the nucleus like a library, and DNA as the book of life stored gently inside. Now, what does this DNA look like? You may have seen pictures of DNA drawn like a twisted ladder. Scientists call this shape a double helix. The word helix just means spiral, and double means there are two strands twisted together. Imagine a zipper. Each side of the zipper has teeth that lock together. In DNA, those teeth are called base pairs, tiny molecules that always join in a special way. One base always pairs with another. A with T and C with G. These four letters, A, T, C, and G, are like the alphabet of life. But instead of writing sentences, DNA uses these letters to build proteins, the tools and machines that your body needs to survive. Here's something amazing. If we could take all the DNA from just one of your cells and stretch it out, it would be about 2 meters long, roughly the height of a tall adult. Yet it's curled up so tightly that it fits inside a space smaller than a speck of dust. And now, think of this. Your body has trillions of cells. Each one has its own copy of this DNA. That means your body is carrying enough DNA to wrap around the earth millions of times, and yet you never notice it. That's the magic of biology. It hides so much wonder in the tiniest places. Now, let's understand what DNA actually does. Think of DNA as a recipe book. It doesn't build the cake. It tells the kitchen how to make it. Your body is the kitchen, and the cake, that's you. DNA gives instructions to make proteins. These proteins are responsible for almost everything in your body. Your muscles, your blood, your skin, even the enzymes that help you digest food. Let's break this down. Proteins are made from smaller parts called amino acids. There are only 20 of them, but they can be arranged in endless combinations, like beads on a necklace. DNA tells your body which amino acids to use and in what order. It's like spelling out a word, one letter at a time. If the order is correct, your body makes a healthy protein. If there's a mistake, even a tiny one, the protein might not work. Sometimes these small mistakes are harmless. Other times they can lead to diseases. That's why DNA is so important. One small change in the code can change a life. But don't worry, Your body is very good at catching these mistakes. It has special systems that scan and repair damaged DNA, like editors fixing a sentence. Now, let's talk about how DNA is stored. Inside your cells, DNA doesn't just float around. It's carefully packed into structures called chromosomes. Humans have 23 pairs of chromosomes, 246 in total. Half come from your mother and half from your father. That means your DNA is a mix of both your parents' instructions. It's why you might have your father's nose, but your mother's smile. Within these chromosomes are smaller regions called genes. You can think of a gene as a specific instruction, like a single recipe in a cookbook. One gene might tell your body how to make a certain protein. Another gene might influence how tall you grow, or how your immune system responds to illness. In total, humans have about 20,000 genes, yet they make up only a small part of your DNA. The rest, scientists used to call it junk DNA, but now we know that even the quiet parts of the code may play hidden roles, helping regulate when and how genes are used. So the DNA is not just instructions, it's a whole orchestra, where some parts sing and others keep silent, all in harmony. Let's pause for a moment. Right now, in your body, DNA is hard at work. Even as you rest, your cells are copying their DNA, fixing it, or using it to build tiny proteins, quietly, gently, continuously. This silent process keeps you alive, moment by moment. It links you to your ancestors and to every other living thing on Earth. Here's a beautiful thought. You share over 98% of your DNA with a chimpanzee, about 85% with a mouse. Even a banana shares about 60% of the same basic code. That's because the core instructions for life are ancient and deeply connected. DNA is nature's way of remembering what works. And when you look at it this way, it's not just a molecule. It's a story, a memory, a bridge across time. The double helix in your cells is the same shape found in plants, animals, even bacteria. It's a pattern written into life itself. And so we end this section with a quiet truth. DNA may be small, but it holds galaxies of meaning. It is the soft whisper that tells your heart to beat, your skin to grow, your thoughts to rise. It is not just what you are made of, it is the reason you are here. Let us now take a closer look, even closer than before. Imagine yourself shrinking gently until you are small enough to stand on the surface of a single strand of DNA. What would you see? You would see a long, twisting ladder, and if you looked more closely, you would notice something even more beautiful. The rungs of this ladder are made from just 4 basic building blocks. These are called bases. Their full names are a bit long: adenine, thymine, cytosine, and guanine. But most people simply call them A, T, C, and G. These 4 letters are the true alphabet of life. That may sound simple, only 4 letters, but simplicity can be powerful. Think about this. The English language uses just 26 letters. Yet from those, we can write poems, stories, and entire libraries. In the same way, these 4 DNA letters, A, T, C, and G, can build all the instructions your body needs. Now let's understand how these letters work. Each letter is a small molecule, and in DNA, these letters always join in pairs. with TC pairs with G-like pieces of a puzzle that fit together perfectly. Imagine beads on a string. The order of those beads matters. In the same way, the sequence of a TC and G determines what kind of message is being written. A short sequence might say, make this protein. A longer one might say, build a heart, or even start hair growth here. This is where genes come in. A gene is a special section of DNA. Think of it like a sentence in a very long book. Each gene carries a specific instruction. Most genes tell the body how to make one particular protein, a kind of tiny worker molecule that keeps the body running. Proteins are not just one thing. Some help build muscles. Others carry oxygen in the blood. Some break down food. And some are even part of your immune system, helping you fight off sickness. So, when a gene sends out its message, it is really saying, make this protein right now and send it where it's needed. Let's pause for a moment. Think about this. Your body has around 20,000 genes. These are scattered across your DNA, much like recipes in a cookbook. And while most of these genes are active in every person, some turn on only in certain places or at certain times. For example, your eye cells use genes that help detect light, but those same genes stay silent in your liver where they are not needed. This ability to turn genes on or off, like flipping a switch, is part of what makes life flexible. It's how the same DNA can create many different kinds of cells in the body. Now here's a small wonder. The total length of your DNA is about 3 billion letters long. That is 3 billion A's, T's, C's, and G's, all lined up in a precise order. If you tried to read it out loud, letter by letter, it would take you over 90 years, reading day and night without stopping. And yet, every cell in your body carries this complete code, a full instruction book hidden inside. Let's make this more visual. Imagine a row of train cars. Each car is a base, an A or a T, a C or a G. Some trains are short, others stretch. On 4 miles, each full train spells out a gene, and these trains are organized on tracks, the chromosomes. Together, they form a massive rail system, running silently inside each cell. It's not just the letters that matter. The spaces between them matter too. Think of a sentence like this. Let's eat, Grandma. Now take away the comma. Let's eat, Grandma. The meaning changes completely. In DNA, small changes can have big effects. A single letter swap can lead to a different protein. Sometimes this causes a visible trait, like eye color. Other times, it may lead to illness, or it might do nothing at all. This is why scientists study the DNA code so carefully. They want to understand which parts are vital, which are flexible, and which are hidden but important. Let's look at an example. There is a gene called HBB that makes part of the hemoglobin protein, the part of red blood cells that carries oxygen. In some people, a single letter in this gene is changed. Just one letter out of 3 billion. That tiny change causes the red blood cells to become stiff and curved, like a sickle. This is called sickle cell disease. The symptoms are serious: pain, fatigue, even organ damage. And all of it starts with one small change in the DNA alphabet. But DNA is not only about disease. It also explains why siblings look alike, and yet different. They share many of the same gene sequences, but not all of them. Some genes are the same. Others are slightly different versions, what scientists call variants. These variants give us our diversity. They help shape our skin color, our height, our risk for certain diseases, and even how we react to certain medicines. This brings us to an important word, the genetic code. The genetic code is the set of rules your body uses to read DNA. It's like a decoder ring. It tells your cells how to translate sequences of letters into real-world proteins. And here's the elegant part. Every three-letter group in DNA, called a codon, stands for one amino acid. Amino acids are the building blocks of proteins. So when the cell reads DNA, it reads three letters at a time: ATG, CCG, TIA. Each set of three builds one small piece of a protein, one codon at a time. The process is slow and steady, precise and graceful, like typing out instructions, letter by letter, never rushing, always exact. So when we speak of DNA as an alphabet, it is more than a metaphor. It is real. A language written in molecules, a sentence written in silence, a message passed from parent to child, from cell to cell, and it is this code that brings your body to life. As we drift toward the next chapter, let's carry this thought. DNA is not just a thing. It is a story being told every moment, a language whispered deep within. And every letter, every line, is part of the quiet song that makes us who we are. Let's return to the quiet world inside your cells. You've already seen how DNA holds the instructions for life. But now we ask, how does the body read this code? How does a cell turn DNA into something real? Imagine you are in a kitchen. You have a recipe book filled with instructions, but you don't want to bring the entire book near the stove. Instead, you copy down the recipe you need and take just that page with you. This is what your cells do. They do not carry the whole DNA instruction book around. This process of copying is called transcription. The word transcription simply means to write something down again, to copy it carefully and clearly. Let's break it down. The cell begins by opening a small part of the DNA, a specific gene it wants to read. Enzymes come in, special helper proteins that act like little machines. One important enzyme in this process is called RNA polymerase. You don't need to remember the name. Just think of it as the cell's copy machine. It moves along the DNA strand and builds a new, matching strand, one letter at a time. But this new copy isn't DNA, it's RNA. RNA stands for ribonucleic acid. It's a close cousin of DNA. In fact, RNA uses almost the same alphabet. A, C, and G stay the same. But instead of the letter T, RNA uses a letter called U, which stands for uracil. So when the DNA has an A, the RNA copy will use AU. The other pairings stay the same. It's like making a translation from one dialect to another, just enough of a change to make the message easier to work with. The result is a strand of RNA that matches the gene the cell wants to use. The M stands for messenger, because that's exactly what it is, a message that carries the code from the nucleus to the rest of the cell. Let's step back for a moment. Imagine the DNA as the blueprint of a building. It contains every detail about structure, size, and function. But when it's time to build something, like a door or a window, the workers don't need the whole blueprint. They need just one page, one part. That's what Mr. NA provides. A small, specific copy. Now let's follow this mRNA message. Once the RNA is complete, it leaves the nucleus, the central chamber of the cell, and moves into the outer area, called the cytoplasm. The mRNA carries its message to a structure called a ribosome. Think of the ribosome as a tiny workbench. It's where the actual building takes place. The ribosome reads the message written in the RNA and begins assembling a protein. It does this by reading 3 letters at a time. These 3-letter sections are called codons. Each codon stands for one amino acid, the small pieces that link together to form a protein. You can imagine the ribosome like someone reading a sentence in Braille. It feels the bumps 3 at a time and uses them to choose the right building block. One codon says, add this amino acid. The next says, add another. This step is called translation because the cell is translating the RNA code into a real physical protein. But let's not rush there yet. We are still in the world of transcription, the careful copying of DNA into RNA. This step is more important than it may seem, because it is here, at the transcription stage, where the cell can decide when, where, and how much of a protein to make. The cell doesn't copy every gene all the time. That would be wasteful. Instead, it selects certain genes at certain times, depending on the body's needs. For example, when you're exposed to sunlight, when you eat a meal, cells in your pancreas might transcribe genes that help produce insulin, the hormone that controls sugar levels in the blood. It's the start of action, a gentle turning of silence into sound. And like all delicate processes, transcription must be precise. If the RNA copy has an error, the wrong protein might be made. Sometimes this can lead to problems, but cells have safety checks, mistakes, editors that remove unnecessary parts. Yes, sometimes the RNA needs editing. When a gene is copied, the first RNA version might include extra parts, regions that don't code for anything useful. These non-coding parts are called intranslations. This editing process is called splicing. Think of it like filming a documentary. You record hours of footage, but only keep the useful scenes. The final film, the edited RNA, is short, clear, and ready to use. Now, let's reflect on the beauty of this step. Transcription is quiet, invisible. It happens every moment, in every cell. And yet, it's what keeps life moving. Without transcription, your body cannot build proteins. Without proteins, nothing functions, not your muscles, your brain, or even your breath. And here's something astonishing. In the human body, cells produce over 100,000 different types of proteins, yet all of them start the same way, with a message copied from DNA. Each time a cell transcribes a gene, it is choosing one page from the book of life, one message from the library of your DNA, and then, softly and silently, it passes that message forward for translation, for building, for living. Let's go back to our earlier metaphor. Imagine a calm scribe in a stone library, transcribing the wisdom of your DNA into messages your body can use. And so, transcription is more than a technical step. It is a bridge between blueprint and builder, between memory and motion, between instruction and expression. As the mRNA leaves the nucleus, it carries not just a code, but a purpose, a whisper from deep within, now ready to become action, and shapes become life. Let us return now to the quiet traveler we left in the last section, the strand of messenger RNA, or mRNA. This tiny ribbon of code has journeyed from the center of the cell, carrying a single message copied from the DNA library. Its destination. Is the ribosome the cell's miniature kitchen? This is where the real work begins. If DNA is the recipe book, and mRNA is the handwritten note copied from it, then the ribosome is the chef's workbench. It reads the note carefully and follows the instructions to prepare something vital, something living. Proteins. Proteins are not just food for muscles. They are the builders, movers, and managers of life itself. Every breath you take, every heartbeat, every thought, all rely on proteins. So how are these proteins made? Let us pause and imagine. Picture the ribosome as a small machine, not much more than a speck inside the cell. Yet it holds a power so immense that all living things depend on it. It reads the mRNA message one piece at a time. Each piece is made of 3 letters. Scientists call this group a codon. Each codon stands for 1 amino acid. Amino acids are the building blocks of proteins. There are only 20 different kinds, but like letters in an alphabet, they can be arranged in endless ways. Some chains are short, others are thousands of units long. Now, picture this. The ribosome reads the mRNA like a sentence, 3 letters at a time, and uses each codon to pick the correct amino acid. A helper molecule called transfer RNA or tRNA brings in these amino acids. Each tRNA carries one specific kind. You can think of tRNA as a delivery truck, bringing the right part to the construction site. The ribosome then links the new amino acid to the growing chain, forming a protein piece by piece. Click. Another codon is read. Click. Another amino acid is added. Slowly, gracefully, the protein begins to take shape. This process is called translation, because the cell is translating the genetic message from the language of bases into the language of amino acids. It is a careful and beautiful dance. Let's break it into steps. First, the ribosome binds to the mRNA. This is like opening the recipe. Next, it reads the first codon, usually a huge. This codon tells the ribosome, start here. A huge always stands for a special amino acid called methionine, which acts as the starting point for most proteins. Then, one by one, tRNA molecules bring the next amino acids, matching them to the mRNA codons. The ribosome forms chemical bonds between them. As the chain grows longer, the protein begins to fold, twisting and bending into a specific shape. And here lies one of the great secrets of biology. A protein's shape is everything. If a protein is shaped correctly, it can do its job. It might carry oxygen, or break down sugar, or send a message to another cell. But if the shape is wrong, if even one amino acid is out of place, the protein may not work at all. This is why the ribosome must read carefully. One wrong letter in the mRNA, one mistake in translation, and the entire protein might collapse. But most of the time, the ribosome gets it right. In fact, your body builds around 100,000 different kinds of proteins, all from the same basic process, all guided by the same 4-letter DNA code. Let's step back for a moment and admire the elegance. Inside every cell, tiny ribosomes are working without rest. A single cell can contain thousands of these protein-making machines. In a human body, that means trillions of ribosomes, all reading, translating, and building all the time. The speed is astonishing. Some ribosomes can add up to 20 amino acids every second, and many ribosomes can work on a single mRNA at the same time, like multiple chefs working from the same recipe. Together, they build the proteins that give structure and strength to your skin, bones, and hair. They build the enzymes that speed up chemical reactions. They form antibodies that help fight infections. Even your mood is shaped by proteins. Serotonin and dopamine, the molecules that affect feelings of happiness and calm, are built and moved around using protein helpers. In this way, proteins are not just parts of you, they are you. Let's return to the protein being built. Once the ribosome reaches the final codon, the stop signal, it knows the recipe is complete. The protein chain is released, but it is not yet ready to work. Most proteins must fold into a very specific 3D shape. Some fold on their own. Others need helpers, special molecules called chaperone proteins. These helpers guide the protein gently, making sure it folds correctly. Once folded, the protein travels to its destination. Some stay in the cell, others are packed into small bubbles called vesicles and sent out into the bloodstream or to other parts of the body. It's like sending a finished tool to a distant workshop or mailing a letter to someone far away. The cell knows exactly where each protein needs to go. This is coordination at the molecular level. It is silent, invisible, and yet it keeps you alive. Now imagine this. Each second, your body creates millions of proteins. Some will last for minutes, others will last for months. Some will help you move, others will help you sleep. And every one of them began with a single quiet step, a strand of RNA drifting toward the ribosome, a message becoming a molecule, a recipe becoming reality. Here is a thought to carry with you. Life does not rely on one giant machine. It relies on trillions of tiny ones working together in harmony. The ribosome is just one, but it is a master builder, assembling the structures of life from strings of meaning. So when we speak of the body, we often name its parts, the bones, the muscles, the organs. But beneath all of these, it is proteins that build, repair, signal, and protect. They are the quiet workers of biology, and every one of them begins in the cellular kitchen, the ribosome. In our next section, we'll follow these proteins further after they are built. Where do they go? How do they find their role? How are they sorted, shaped, and delivered across the body? We will explore the secret pathways that proteins take, the cellular highways that help them reach their destination. But for now, rest in the wonder of this one fact inside you. Right now, millions of proteins are being made carefully, quietly, and with astonishing precision. We now arrive at the moment where function begins. The protein has been built, folded, shaped, delivered. But what does it do next? Proteins are not just passive shapes floating in the cell. They are workers, they are tools, they are defenders, builders, messengers, and cleaners. From the tiniest reaction to the largest system, proteins are behind it all. Let's begin with a soft example. Imagine a small paper cut on your finger. You barely feel it, but within moments, your body responds. Cells rush to the site. Blood begins to clot. Tissue begins to rebuild. All of this happens because of proteins. One protein helps your blood cells stick together and form a clot. Another guides immune cells to clean the area. Still others rebuild the skin from the inside out. This is not magic. It is biology in motion, quiet, exact, and always prepared. Proteins that help repair and build are often called structural proteins. One of the most famous is called collagen. Collagen is like scaffolding inside the body. It holds tissues together. It gives your skin strength. It supports bones, tendons, and even your eyes. Another structural protein is keratin. It forms your hair, your nails, even parts of your skin's outer layer. Tough but flexible, keratin protects you from the outside world. Now consider another role: movement. Muscles contract when long fibers slide past one another. These fibers are made of proteins like actin and myosin. They grip, they pull, they release, over and over again. Every time you move your fingers, blink your eyes, or take a breath, proteins are behind the motion. Let's go deeper now, inside your bloodstream. Here we meet one of the most vital proteins of all, hemoglobin. Hemoglobin lives inside red blood cells. Its job is simple, yet powerful: carry oxygen. It binds oxygen in your lungs and lets go of it in your tissues. Without hemoglobin, oxygen could not travel through the blood. But hemoglobin does more than carry. It adjusts. It senses when oxygen is low and releases more. It shifts its shape to respond to changes in pH and temperature. In this way, it is both a carrier and a responder. There are proteins that protect you, too. Some are called antibodies. Antibodies are part of your immune system. They float through your blood, watching quietly. When they detect something foreign, like a virus or harmful bacteria, they attach to it. This attachment marks the intruder for destruction. Each antibody is shaped to match just one threat, like a lock that fits only one key. When a match is found, it signals other immune cells to take action. Now let's visit digestion. When you eat food, your body breaks it down into smaller parts: proteins into amino acids, fats into fatty acids, sugars into simple forms like glucose. To do this, your body uses enzymes. Enzymes are proteins that speed up chemical reactions. They do not get used up. They are reusable helpers. For example, when you eat bread, an enzyme in your saliva called amylase starts breaking down the starch into sugar. Later, in the stomach, another enzyme called pepsin breaks protein chains into smaller pieces. Without enzymes, these reactions would be far too slow to sustain life. Some enzymes are found in your liver. There, they help break down drugs, alcohol, or toxins. They prepare these chemicals so they can be removed from the body safely. And again, these enzymes are made of proteins. Let's pause and reflect on something remarkable. Proteins are made using amino acids, the very same ones we get by eating protein-rich foods. This is a full circle. The proteins in your food become the amino acids your body uses to build new proteins. This recycling is efficient and elegant. Nothing is wasted. Now move your attention to communication. Another vital job proteins handle. Hormones are the body's messengers. And while not all hormones are proteins, many of them are. Insulin is a protein hormone. It helps regulate the amount of sugar in your blood. When you eat, your blood sugar rises. In response, your pancreas releases insulin. Insulin tells your cells to absorb the sugar and store it or use it for energy. Without insulin, sugar builds up in the blood, leading to serious health problems. Another example is growth hormone, which, as the name suggests, helps control growth in children and teens. It also helps repair tissue throughout life. Even your mood is influenced by protein-based messengers. Endorphins are proteins that help reduce pain and create feelings of calm or happiness. After exercise or during moments of stress relief, endorphins are released, easing discomfort and lifting mood. Proteins also act as gatekeepers. On the surface of every cell, there are protein channels, small openings that let things in or out. Some allow sodium or potassium ions to pass through. Others help sugars or water move. These channels are very specific. They open only when needed and only to the right substances. In this way, proteins help maintain balance inside the cell. And there's more. Proteins are involved in vision opsins. In the eye, respond to light. In hearing, proteins in your ear vibrate with sound waves. In memory, proteins in the brain help form new connections between nerve cells. Everywhere you look, proteins are working. Some stay in one place for a long time. Others are made, used, and destroyed quickly. Some are large and complex. Others are small and simple. But each one is part of the quiet machinery that keeps you alive. Let's return to the image of the ribosome. It reads the RNA, builds the protein, folds it carefully, sends it out into the world. The protein then finds its place. Maybe it becomes part of your skin, or an enzyme in your mouth, or a carrier in your blood. Each one has a purpose. Each one answers a need. This is the story of life on a molecular scale, and it reminds us behind every heartbeat, breath, and thought, proteins are at work. They are the tools of life, invisible, tireless, precise. As we move forward, we'll ask another question. Where do all these instructions come from in the first place, and how do they get passed on from one generation to the next? To answer this, we must return to the beginning, to the passing of genes from parent to child, and to the way inheritance shapes not only how we look, but how we function. But for now, let the quiet work of proteins settle in your thoughts. They are within you, always, working softly, building silently, and keeping life in motion. Imagine a vast city at night. The buildings stretch into the horizon. Some windows glow softly, others remain dark. Each light, each room serves a different purpose. A bedroom, a kitchen, and within each cell, genes are the rooms. But not all rooms are lit at once. Some are active, some are silent. This is gene expression. Every cell in your body has the same DNA. That means the same full set of genes, from your skin to your brain to your heart. But cells are not all the same. A skin cell makes protective proteins. A heart cell beats rhythmically. How is that possible if they all share the same DNA? The answer is this. Each cell turns on only the genes it needs, and it turns off the rest. This selective use of genes is what makes each cell type unique. It is like choosing which lights to turn on in a building, depending on what job is being done. Let's look at how this works. First, a cell needs to read a gene in order to use it. The first step in that process is called transcription. Want to read that gene? Then it places a kind of block, a signal that prevents the reading from starting. This is how genes are turned off. A signal placed at the start of a chapter. If the bookmark says read, the cell reads it. If the bookmark says stop, the cell skips it. These bookmarks are called regulatory elements. They sit just before the gene begins. Some attract proteins that start the reading. Others attract proteins that stop it. The proteins that help turn genes on are called transcription factors. They are like little switches. Together, they control which genes are used and when. But who controls the switches? That's where it gets even more fascinating. Some signals come from inside the cell. These may be based on age, growth, or what stage the cell is in. For example, an embryo will turn on genes for early development, but turn them off later in life. Other signals come from outside the cell: food, temperature, stress, hormones, even sunlight. These outside signals can tell a cell which genes to turn on. This is why, for example, your skin makes more pigment when exposed to sunlight. The sun sends a signal. The signal turns on pigment-producing genes. Gene control is not random. It is careful, precise, and layered. In fact, genes can be turned on strongly, softly, or only briefly, like a volume knob, not just a switch. Let's go deeper. This word means above genetics. It refers to changes that affect gene activity without changing the DNA letters themselves. One common type of epigenetic change is called DNA methylation. Cell not to read that gene. Another type involves the structure of the DNA itself. DNA is wrapped around small protein balls called histones. When the wrapping is tight, the gene is hidden and cannot be read. When it's loose, the gene is exposed and can be used. So, the cell can tighten or loosen the wrapping, controlling which parts of the DNA are open for reading. This kind of control allows cells to adapt. Imagine you are exercising. Your muscle cells need more energy. They respond by turning on genes that help make more mitochondria, the parts of the cell that produce energy. Or consider a time of infection. Would waste energy on other tasks. The control is fast, temporary, and reversible. But sometimes gene switches stay fixed for a long time. This happens during development. As an embryo grows, different cells are assigned different roles. Some become nerve cells, others become skin. These decisions are often locked in with many genes permanently turned off to keep the cell focused on its job. This is how a nerve cell stays a nerve cell, even decades later. To turn one cell into another type, we must first undo these permanent switches, a task that is complex and delicate. Gene control is also involved in diseases. In cancer, for example, the genes that normally stop cells from growing may be turned off. At the same time, genes that cause growth may be turned on too strongly. This imbalance leads to uncontrolled cell division. Understanding gene regulation helps us find new treatments. Some drugs work by turning genes back off or on, gently nudging the switches into the right position again. There are also proteins called repressors. The road until the repressor is removed, the gene stays silent. On the other hand, activators help open the DNA and make it easier to read. Think of all these players: transcription factors, repressors, activators, histones, and methyl tags as part of a giant control panel. Each one adjusts a setting. The goal is balance. Not too much, not too little. Just right for the needs of the cell. Now step back and reflect. Inside each of your cells, millions of them genes are being chosen every moment. Some turned on, some turned off. You do not feel it, you do not hear it, but it shapes every part of you. Your eye color, your blood type, your tolerance to caffeine, even parts of your personality, all are influenced by how your genes are used, not just which ones you have. And many of these decisions were made early in life, some even before birth. So while your DNA is fixed, your gene expression is not. It is flexible, responsive, alive. In the next section, we will explore how these gene patterns are passed on, from parent to child, from one generation to the next. But for now, rest with this thought. You are not just built from DNA. You are shaped by when and how that DNA is used. Like a symphony, where not every instrument plays at once, but together they form a harmony called life. Picture a handwritten letter, neat lines of script, each word carefully placed. Now imagine a small smudge appears. A single letter is changed. Maybe the meaning stays the same. Maybe it shifts. Maybe it's no longer understood. This is how a mutation begins. Inside every cell of your body, DNA carries the instructions for life. Billions of letters, precise, ordered. Yet, sometimes, a tiny error slips in. This change in the DNA sequence is called a mutation. It can be as small as switching one letter, like replacing an A with a G, or it can be as large as deleting a chunk of the code, or even copying something twice by accident. Some mutations happen quietly. Others cause a ripple that changes the way a cell behaves. Some are passed on. Some are corrected before they cause harm. And some, though rare, help life evolve into something new. Let's begin by looking at where mutations come from. One source is simple: mistakes during cell division. Every time a cell copies its DNA, it makes a full duplicate. This means copying over 3 billion letters, one by one. Even though the cell uses special tools to check for errors, sometimes one slips through. Think of it like typing an essay without spell check. One wrong letter here or there. Another source is radiation. Ultraviolet light from the sun, for example, can damage DNA in skin cells. That's why sunburns, especially repeated ones, can increase the risk of skin cancer. The UV rays cause breaks or twists in the DNA strand. There are also chemical causes. Certain toxins, pollutants, or cigarette smoke contain substances that can sneak into the DNA and change its structure. Some chemicals make the DNA harder to copy. Others break it completely. Then there are viruses. Some viruses insert their own genetic material into a host's DNA. If this material lands in the wrong spot, it can disrupt the host's genes, turning normal cells into dangerous ones. Finally, some mutations are simply inherited. You were born with them, passed down from your parents through the egg or sperm. These inherited mutations can explain why some diseases run in families, but not all mutations are harmful. In fact, many are neutral. They don't change the function of the gene, or they occur in parts of the DNA that aren't actively used. And some are even helpful, giving rise to genetic diversity. Think about how different we are from one another. Eye color, height, hair texture, skin tone, the shape of our noses, the way we digest certain foods. Much of this comes from slight differences in our DNA, many of which began as mutations long ago. Over time, helpful mutations can spread in a population. This is one of the engines of evolution. Let's take an example. Thousands of years ago, most humans lost the ability to digest milk after childhood. But in some people, especially in Europe and parts of Africa, a mutation allowed the gene for milk digestion to stay active into adulthood. This small change offered a survival advantage. People who could digest milk had more food options during times of famine. The mutation spread today. That's why many adults in those regions can drink milk without trouble, while others around the world still become sick from it. This shows that mutations can lead to adaptation. They are random, but natural selection helps decide which ones stay. Now let's explore the different types of mutations. The smallest is called a point mutation. A single letter is changed. Like writing cat as bat, sometimes this causes no problem. Other times, it changes the meaning of the entire protein. Another type is a deletion, where one or more letters are removed, where extra letters are added in. These can shift the entire reading frame of the gene. It's like removing a word from a sentence and suddenly losing the whole meaning. For example, take this sentence: the big red dog ran. Now delete one letter: THB, Iger, ET, ogre, an. The sentence is no longer readable. That's what can happen in a frameshift mutation. Some mutations duplicate entire segments of DNA, repeating the same code over and over. Others invert a segment, flipping it backward. Larger mutations can even involve swapping pieces between chromosomes, a kind of genetic rearrangement. Not all of these cause harm, but some do. Let's look at sickle cell disease. This condition comes from a single letter change in a gene that makes hemoglobin, the protein that carries oxygen in red blood cells. Because of the mutation, the red blood cells become stiff and shaped like sickles, curved instead of round. This causes them to block blood flow and break more easily. But here's something fascinating. The same mutation that causes sickle cell disease also offers protection against malaria, a deadly disease caused by a parasite that infects red blood cells. People who carry just one copy of the sickle gene usually don't get sick, and they are more resistant to malaria. That's why this mutation has stayed in certain populations where malaria is common. So even harmful mutations can carry hidden benefits. Another example is cystic fibrosis, a genetic disorder that affects the lungs and digestive system. It's caused by a mutation in a gene that helps control salt movement in cells. Without proper salt balance, thick mucus builds up in the lungs, making it hard to breathe. Understanding this mutation has helped scientists develop new treatments, some that target the faulty protein directly and improve quality of life for those affected. Science continues to uncover more examples where knowing the mutation leads to better care. Modern tools like genetic testing allow us to find these changes early. In some cases, we can even prevent symptoms from developing. In the future, gene editing may let us correct mutations at the source, using tools like CRISPR, a technology that acts like molecular scissors, cutting and replacing faulty parts of the DNA. But this field is still new and filled with important questions both scientific and ethical. Should we edit genes in embryos? What counts as a disease, and what is simply variation? Who decides which traits are desirable? These are deep questions, and science is still exploring the answers. What we do know is this: mutations are part of life. They are not always mistakes. Sometimes they are nature's way of writing a new sentence in the book of life. Sometimes they whisper change, and other times they shout. But whether helpful, harmful, or neutral, mutations shape every species on Earth. They drive evolution. They shape health. They create the endless variety of living things. And they remind us, even the smallest change can have great meaning. In our next section, we will explore how the journey of inheritance continues, how parents pass their DNA to children, and how the traits we carry may echo far beyond our own lives. For now, let the idea settle gently. Even a single letter can change the story. Look at your hands, the shape of your fingers, the curve of your nails, maybe even the way they rest when relaxed. These features were shaped long before you were born. Inside every person, there is a quiet thread that connects the past to the present, a line that weaves through parents, grandparents, great-grandparents, and beyond. That thread is DNA, passed on from one generation to the next. This passing down of DNA is called inheritance. It is how we receive certain traits from our family. Some traits are easy to see: eye color, hair texture, skin tone, height. Others are hidden deep inside, like blood type, how your body handles certain medicines, or whether you're likely to develop a disease later in life. Let's begin at the beginning. When a baby is made, it receives half of its DNA from each parent. One half comes from the egg. The other half comes from the sperm. Together, they create a full set, a brand new combination of genetic instructions. Think of it like mixing two decks of cards. Each parent contributes their own set, but the way the cards shuffle creates something unique. That's why you might have your mother's eyes and your father's nose, or your grandfather's curly hair and your aunt's dimples. Some traits appear to skip a generation. Others show up in unexpected ways. Now let's explore how these traits are passed down. Inside the DNA, specific sections are called genes. Each gene carries instructions for one feature, like weather. Your hair will be straight or wavy. But here's something important. Most traits are controlled by pairs of genes, one from each parent. Some gene versions are dominant, some are recessive. A dominant gene is like a loud voice. It only takes one copy for its trait to show. A recessive gene is quieter. It needs two copies, one from each parent, to be seen. Imagine this. There is a gene that controls whether someone can roll their tongue. The version that lets you roll your tongue is dominant. The version that doesn't is recessive. So, if a person has one dominant and one recessive version, they will still be able to roll their tongue. Only if they get two recessive copies, one from each parent, will the trait be hidden. Another common example is earlobe shape. Earlobes are usually dominant. Attached earlobes are usually recessive. Again, the dominant form only needs one copy to appear. Now let's look at eye color. It's more complex than it seems. Many people think brown eyes are dominant and blue eyes are recessive. This is mostly true, but eye color is actually controlled by multiple genes, not just one. This is called a polygenic trait, meaning many genes work together to decide the final outcome. That's why a child can have green or hazel eyes, even if their parents have different eye colors. In fact, most of our features are polygenic, like height, skin color, and even intelligence. These traits do not follow simple, dominant, and recessive rules. They are shaped by a blend of many gene versions, along with environmental factors, like food, sleep, or sunlight. Let's now visit the idea of family trees. In genetics, a family tree is called a pedigree. It's a diagram that shows how a trait is passed down through generations. Doctors and scientists use pedigrees to understand inherited diseases. For example, imagine a disease caused by a recessive gene. If both parents carry one copy of the gene but don't show symptoms, each of their children has a 25% chance of inheriting 2 copies and being affected. This is how conditions like cystic fibrosis or Tay-Sachs disease are passed on. In contrast, diseases caused by dominant genes only require one faulty copy to show symptoms. These might include Huntington's disease or Marfan syndrome. Pedigrees help doctors see patterns: who was affected, who was a carrier, and what risks future children may face. Now let's talk about sex-linked traits. These are traits tied to the X and Y chromosomes, the pair that decides whether someone is biologically male or female. Females usually have 2 X chromosomes. Males have 1 X and 1 Y. Some genes are only found on the X chromosome. If a male inherits a faulty gene on the X, there is no backup copy to protect him. That's why certain conditions, like hemophilia or color blindness, are more common in males. They only need one faulty X gene to be affected. Females with 2 X chromosomes are more likely to be carriers, meaning they have the gene but may not show symptoms. Let's slow down and reflect. Inheritance is not just about traits we can see. It's about risk and resilience. It's about how we respond to medicine. It's about what illnesses we may carry quietly, even if we never feel them. And it's also about beauty. The shape of a smile that matches your uncle's. The sound of laughter that echoes your grandmother's. Now let's step back. Each human shares over 99.9% of their DNA with every other human. Only 0.1% makes us different. That tiny fraction holds our eye color, blood type, and hair texture. It carries the blueprint for our metabolism, our skin, and our voice. And within families, we share even more. Parents and children share about 50% of their DNA. Siblings also share around 50%, though each combination is slightly different. Grandparents and grandchildren share about 25%. Cousins share around 12.5%. These percentages explain why we look alike, sound alike, or even move alike. Genetics is the language of connection, but it doesn't decide everything. Our lives are shaped by both nature and nurture, by genes and by environment. The foods we eat, the air we breathe, the people who raise us. These experiences interact with our genes to shape the full picture of who we are. Let's take one final example: identical twins. They start with the exact same DNA, yet over time they may grow to look and behave differently. Why? Because their environments are not exactly the same. One may eat differently. One may have a different stress level or sleep pattern. These experiences can affect which genes are turned on or turned off, a field called epigenetics, which we'll explore soon. So, inheritance is not a rule book, it's a guide, a map, and like any map, it has room for many different paths. The family tree is more than just a chart. It's a living history, a record of where we came from, and a glimpse of where we may go. And while genetics explains much about us, it leaves room for something more: choice, chance, and change. In the next section, we'll step into that space between genes and environment, where the body reads not just what's written, but also when, how, and why. That world is called epigenetics, and it reminds us that the story of life is not fixed, it's dynamic. Let's turn the page together. Imagine holding a book that contains the story of your life, not just your past, but clues about your future. A book so detailed that every cell in your body keeps a copy. That book is your DNA. And today, scientists have learned how to read it line by line. This new ability has changed how we see ourselves. It has also opened doors to powerful new tools, from understanding disease to reshaping medicine. This field is called genomics, the study of all the genes in the body. Let's gently step into this world. In the year 2003, something incredible happened. After more than a decade of work, scientists completed the Human Genome Project. For the first time, they had read and recorded the full sequence of human DNA. That's more than 3 billion letters, a long chain of a T, C, and G. This achievement was like mapping the surface of a new planet. Suddenly, we had a complete guide to the genetic blueprint of humans. But reading the letters was just the beginning. Now, scientists wanted to understand what each part of the code did. Which sections controlled eye color? Which ones were involved in cancer or diabetes? To answer these questions, new tools were created. One of the most important tools is DNA sequencing. This is the process of reading the exact order of genetic letters in a sample. Modern machines can now do this in just a few hours. In the past, it took years. Thanks to this, a person today can send a tiny tube of saliva to a lab and receive a report on their ancestry, traits, and health risks. Companies like 23andMe and Ancestry DNA have made this technology widely available. Millions of people have now seen pieces of their genetic story, but this information comes with care. DNA tests can reveal sensitive truths. They might uncover family relationships or show a person has a higher chance of getting a disease, like Alzheimer's or breast cancer. That's why ethics are so important in genetics. Ethics is the study of right and wrong. In genetic science, it asks, who should see your DNA data? Can insurance companies use it? Should you be told about risks you cannot change? Laws like the Genetic Information Non-Discrimination Act, or GINA, were created to protect people. In the United States, they say your genetic data cannot be used against you by health insurers or employers. Still, scientists and doctors move carefully because DNA is not just data, it's deeply personal. Let's now explore another modern wonder: gene editing. Imagine being able to fix a spelling mistake in the book of life. That is what gene editing tries to do. The most famous tool for this is called CRISPR, spelled CRISPR. It stands for clustered, regularly interspaced, short palindromic repeats. But you don't need to remember that. Just think of CRISPR as a pair of tiny scissors that can cut and change DNA. With CRISPR, scientists can remove a faulty gene or insert a healthy one. This opens the door to powerful possibilities. One day, we might use it to cure inherited diseases like sickle cell anemia or muscular dystrophy. Some scientists are already trying this in clinical trials with real patients, but CRISPR also raises deep questions. Should we edit the genes of unborn babies? What if people try to create so-called designer children with chosen traits like height or intelligence? Where is the line between healing and enhancement? These are not simple questions, and around the world, different countries have different rules. Some allow gene editing only in adults and only for serious disease. Others forbid editing embryos entirely. So the future of this technology must be guided by both science and wisdom. Another amazing tool is gene therapy. This is when doctors give someone a healthy gene to replace one that isn't working. Gene therapy doesn't cut the DNA like CRISPR does. Instead, it adds a working copy into the body, usually using a harmless virus as the delivery tool. In some cases, this has helped children who were born without functioning immune systems. Others, with blindness or rare blood disorders, have also seen hope. These are small steps, but powerful ones. Let's pause here. All of these tools, DNA testing, CRISPR, gene therapy, are part of a growing field called personalized medicine. This means using a person's genes to choose the right treatment for them. For example, some cancer drugs only work if a patient has a certain gene version. Others might cause side effects in one person, but not in another. By studying someone's DNA, doctors can tailor the medicine, not too strong, not too weak, just right. This is sometimes called precision medicine. It's like giving a key that fits only one lock, made for that individual body. The future may bring gene-based vaccines, new cancer treatments, or even ways to slow aging. But again, the journey must be careful, because with great power comes great responsibility. And now, let's talk about a field that's closely tied to genes, epigenetics. This field explores how the environment can change how genes behave, without changing the letters themselves. It's like placing bookmarks in your DNA. Some pages become easier to read, others get hidden. Things like diet, stress, and sleep can influence which genes are turned on or turned off. This reminds us that while we cannot change our DNA, we can still shape how it works. Even twins with identical DNA can grow up to be very different based on their life experiences. So, while DNA holds the instructions, the body still chooses how to read them, like a conductor leading a song. Let's close this section with a gentle summary. We now live in a time when the code of life is no longer a mystery. We can read it, test it, and sometimes even rewrite it. We can trace ancestry, diagnose disease, and dream of cures once thought impossible. But we must also walk with care, because DNA is not just a tool, it is a thread that connects biology to identity and science to soul. In the next section, we will step back from the lab and microscope. We'll ask a deeper question. Why does all this matter? And how does understanding genetics help us heal not just the body, but also our view of what it means to be human? let's begin that quiet reflection in the final chapter. Every cell in your body holds a library. That library carries not only your story, but echoes of the journeys taken by your ancestors. Woven into your DNA are quiet records of migration, survival, and change. Genes are more than blueprints for making proteins. They are also storytellers, silent witnesses to the environments our species has passed through, adapted to, and overcome. In this section, we gently explore a few of those stories, not through names or dates, but through the genes that shaped human life across the globe. Let's begin in Africa, the place where human life first began. In certain regions of Africa, a surprising genetic twist appears, the sickle cell trait. This is a small change in the gene that makes hemoglobin, the protein in red blood cells that carries oxygen. For most people, red blood cells are smooth and round. But in those with the sickle cell trait, the cells become shaped like tiny crescents. Why would this shape persist over time? Because it offers protection. In places where malaria is common, especially in parts of sub-Saharan Africa, carrying just one copy of the sickle cell gene helps protect against this deadly disease. Malaria is caused by a parasite that enters red blood cells, but when the cells are misshapen, the parasite struggles to survive. This is an example of natural selection, where a trait that helps survival becomes more common. It comes with a cost. If someone inherits 2 copies of the sickle gene, they can develop sickle cell disease, which is painful and dangerous. But the balance of risk and protection shaped the population. Now, let's travel north to Europe. Here, we find a different kind of genetic story. This one is about milk. Most mammals, including humans, stop drinking milk after they are weaned, and they lose the ability to digest lactose, the sugar in milk. But in some parts of Europe, something unusual happened. A genetic mutation appeared that kept the lactase enzyme active into adulthood. Lactase is what breaks down lactose. With this gene, people could drink milk for life. This trait is called lactase persistence. It became especially helpful when farming and dairy animals became part of human life. Milk offered calories, hydration, and nutrition, all in one. Over generations, the people who could digest milk had a small survival edge, and so the gene spread. Today, around 90% of people in Northern Europe can digest milk as adults. But in East Asia, where this mutation did not spread, most adults are still lactose intolerant. It's a reminder that there is no single normal gene. What is helpful in one place may be rare or unnecessary in another. Now let's move east to Asia. Here, the genetic story becomes even more ancient. As humans left Africa and began to populate the world, they sometimes met other human-like species. One of them was the Neanderthals. These were close cousins of modern humans, living in parts of Europe and Western Asia for many years. It was believed they disappeared without leaving a trace. But then scientists looked closely at DNA. To their surprise, they found that many people today, especially those with European or Asian ancestry, carry small pieces of Neanderthal DNA. These pieces make up about 1 to 2% of their genomes. They are not just fossils in our genes. Some still affect the way the body works. For example, certain Neanderthal genes influence how the immune system responds to infections. Others affect skin tone, hair texture, and even the body's reaction to sunlight. It seems that, long ago, when modern humans met Neanderthals, they didn't just compete, they also mingled and shared genes. This story of blending continues in many parts of the world. In Tibet, for example, people live at extremely high altitudes, often above 13,000 feet. At this height, oxygen levels are much lower than at sea level. Most people would struggle to breathe. Their bodies would react by making more red blood cells, which thickens the blood and can lead to problems. But the Tibetan people have a special genetic variant that allows their bodies to function efficiently with less oxygen, without producing excess red blood cells. This variant is believed to have come from another ancient human group called the Denisovans, who lived in Asia before disappearing. Once again, the genes that help people survive in specific environments have been quietly passed down. Let's pause and reflect. Each of these examples, sickle cell in Africa, milk digestion in Europe, high-altitude genes in Tibet, shows how humans adapted to local challenges. The Earth shaped our biology just as we shaped our cultures and tools. Now, with modern science, we can read these changes. Genetic testing lets people trace where their ancestors came from. By studying small patterns in the code, scientists can map ancient migrations, such as how people reached the Pacific Islands, or how Native American ancestors crossed into the Americas. DNA becomes a compass, pointing not just to where we are now, but where we began. But it's important to say this gently. No gene defines a person's worth. No ancestry makes someone better or worse. Genes tell stories of survival, not superiority. In fact, most of our DNA is shared. You and a person from another continent with different skin color, language, and culture still share more than 99% of your genetic code. We are more alike than we are different. And within that tiny percent of variation, we find stories of deserts crossed, winters endured, and foods discovered. This is the poetry of population genetics. Let's explore one more quiet detail. Sometimes genes that once helped survival now become health risks. For example, in ancient times, storing fat was a helpful trait. Food was scarce, and those who stored energy well could survive longer. But today, in a world where food is more available, the same genes may contribute to obesity or diabetes. This mismatch between old adaptations and modern life is called evolutionary lag. It reminds us that our biology still carries echoes of an earlier world. So when we see a gene, we are not just seeing a molecule. We are seeing a traveler, a survivor, a silent companion that has moved across lands, centuries, and families. Our genes are records, not of nations or borders, but of environments, decisions, and small chances that shaped life. As we near the final chapter of this story, we step into reflection. What does it mean to carry such a history inside? And how can this knowledge help us build a future where science and humanity walk side by side? Let us begin that gentle closing where genes meet meaning. We often think of genes as instructions for life, quiet blueprints passed down from parent to child. But what happens when something in the code goes wrong? Imagine a single letter in a long sentence being misread. If the word still makes sense, the sentence may go on as normal. But if that one letter changes the word entirely, the meaning can shift or vanish. In our bodies, This is what can happen when a gene mutates. Sometimes it's harmless. Other times it's life-changing. Let's begin gently. Inside each of your cells, over 20,000 genes are working. Most of the time, they perform quietly, making proteins, giving signals, helping cells grow and divide. But these systems are delicate, and like any system, they can be disrupted. A mutation is a change in the DNA code. It can happen by accident, during cell division, or from exposure to chemicals, sunlight, or radiation. It can also be inherited, passed down from a parent who carries the change in their own cells. Most mutations are small. A single letter in the code changes, or a part of the gene is deleted or repeated. But even small changes can have big effects. Let's look at a few real examples. One of the most well-known is cystic fibrosis. This condition is caused by a mutation in a gene that helps control the movement of salt and water in and out of cells. In people with this mutation, the body makes very thick, sticky mucus, especially in the lungs and digestive system. Breathing becomes difficult. Infections are more common. Food is harder to absorb. Cystic fibrosis is a recessive condition, meaning a person needs to inherit two faulty copies of the gene, one from each parent, to be affected. Carriers who have only one copy usually show no symptoms, but can pass the gene on. Another example is Huntington's disease. This is a progressive condition, meaning it gets worse over time. It's caused by a repeated stretch in a particular gene. People who inherit this repeat may begin to lose control of their movements, memory, and mood, often in midlife. Unlike cystic fibrosis, Huntington's is dominant. A person only needs one changed copy of the gene to develop the disease. This brings us to a delicate idea: genetic risk. Not all conditions caused by genes appear early, and not all gene mutations mean someone will get sick. Some genes increase the risk of illness, but do not guarantee it. One example is the BRCA1 and BRCA2 genes. These are involved in repairing DNA damage. When these genes carry certain mutations, the body becomes less able to fix mistakes in cell division. Over time, this can lead to certain cancers, especially breast and ovarian cancer. But having a BRCA mutation does not mean a person will get cancer. It means their risk is higher than average. This is where science meets care. With genetic testing, people can learn about their risks early and take steps to monitor their health or reduce that risk. Some may choose more frequent screenings. Others may choose preventive surgeries or medicines. Each decision is personal, and science offers the knowledge, not the command. Let's pause and ask, can damaged genes be repaired? This is the heart of genetic medicine, a growing field filled with both hope and caution. One major breakthrough is gene therapy. This is a technique where a healthy gene is inserted into the body to replace or help a faulty one. It's still new and used mostly for rare diseases, but early results are promising. For example, some children born with severe immune system problems, once called bubble boy disease, have been treated with gene therapy by adding a correct copy of the missing gene into their stem cells. Doctors have helped these children develop working immune systems. Another frontier is CRISPR, a tool that can edit genes with great precision. Imagine having a pencil that could erase a single letter from a sentence and replace it with the right one without damaging the rest of the paragraph. CRISPR holds that promise. It has already been used in laboratory settings to correct genetic diseases in animals and in human cells. One of the first clinical uses corrected sickle cell disease by turning on a backup gene for healthy blood. But with this power comes responsibility. Editing the code of life is not just a technical challenge, it's an ethical one. Doctors and communities must ask, should we use gene editing only to treat illness, or also to enhance traits? How do we protect against misuse? And how do we ensure that access to these treatments is fair and not just for the privileged? These are not easy questions, but by asking them slowly, openly, and wisely, we stay grounded in both science and humanity. It's also worth remembering, not all genetic changes are bad. Some create diversity, some offer protection, some may be neutral, but may become important in the future. In the past, the sickle cell trait protected against malaria. In the future, another mutation may help resist a new disease. Our genes are not fixed statues. They are living scripts shaped by time, chance, and environment. As we close this chapter, let's revisit a simple truth. When genes go wrong, it can bring illness, but it can also bring insight. Every rare condition teaches scientists more about how the body works. Every child born with a genetic condition challenges us to be more compassionate, more curious, and more creative. And through these challenges, medicine evolves, from managing symptoms to replacing proteins, from supporting families to editing the DNA itself, from blind guesses to targeted treatments. This is the quiet revolution of genetic medicine. We no longer see illness as a mystery. We now see it as a story, written in code, waiting to be read, understood, and, sometimes, rewritten. We do not blame the code, we learn from it. And in doing so, we come closer to healing, not just the body, but the way we understand life itself. Now, as we gently prepare to leave the world of genes, one final section awaits, a reflection on why all of this matters. Because in the end, the story of genetics is not just about molecules, it's about life, it's about care, it's about hope. Let's pause for a quiet moment. We've journeyed deep into the code of life. We've traced how genes guide the building of bodies, how they help cells speak, how they shape traits, from eye color to how we process medicine. But why does it matter? Why should we care so deeply about this invisible script written inside our cells? The answer is simple and yet profound. Because understanding our genes helps us understand ourselves, not just as individuals, but as families, as communities, and as a species. Let's begin with identity. You are made of about 37 trillion cells. Each cell carries the same basic instruction manual, but how those instructions are read and when makes you who you are. Why do you resemble your parents, or your siblings, or maybe your grandmother? Why do some families have more twins? Why do some people need more sleep, or can digest milk into adulthood, while others cannot? The answers live in your DNA. Genes don't decide everything, far from it, but they give you a starting place, your height, your hair type, even how you respond to certain foods or medicines. These patterns come from combinations of genes, some active, some quiet. And then there's the environment, where you live, what you eat, how you move. and what you breathe. All these factors interact with your genes. This is the science of epigenetics, the study of how environment affects gene activity without changing the DNA code itself. It's as if genes are instruments in an orchestra. Epigenetics is the conductor, deciding which ones to play and when. This brings us to a powerful idea that even if we inherit certain risks, we can sometimes change the outcome. For example, if someone carries a gene that raises their chance of heart disease, they might still stay healthy by eating well, moving more, and reducing stress. In other words, genes are not fate. They are guides, not guarantees. That idea brings comfort to many. It also empowers doctors to help people earlier, before symptoms appear. With genetic testing, some diseases can be predicted or even prevented. This is called predictive medicine. Doctors can screen for certain cancers or adjust drug choices based on a person's genetic makeup. This is called personalized medicine. Instead of guessing, science is beginning to tailor care to each person's unique blueprint. Just like a custom-made shirt fits better than one bought off the shelf. But this power also raises questions. Should everyone get their DNA tested? Who should have access to the results? What if the results bring fear or uncertainty? And how do we make sure that new technologies, like gene editing, are used wisely and not for vanity or profit? These are not just science questions, they are ethical questions, and they remind us that genes are not just about biology. They are about values, choice, responsibility. Imagine a future where rare diseases are cured before birth, where cancers are caught years earlier, where damaged genes are repaired like typos in a sentence. It is not science fiction. It is already beginning. But we must walk slowly, because with every discovery comes great responsibility. Genes connect us. That is one of their quiet truths. You share over 99.9% of your DNA with every other human on Earth. That's right. Less than 0.1% makes you unique. That tiny difference shapes your face, your voice, your rhythm. But the rest of your code, the overwhelming majority, is shared. It is passed down from ancient people who lived in Africa, Asia, Europe, and beyond. It is shaped by migration, weather, survival, and love. It means we are far more alike than we are different. We may speak different languages, eat different foods, follow different beliefs, but inside, in the code of life, we are family. Understanding genetics teaches us humility and unity. It shows us that illness can come from within, but so can healing. That variation is not a flaw, it is strength. That differences in skin, blood type, or disease risk are part of a shared story, not a reason to divide. Let's take a final metaphor. Imagine a vast library. Each book is a person. Each page is a gene. Some books are old, some are new. Some pages are marked or folded or rewritten over time. Together, they form the story of life on Earth. Our one book in that library. Beautiful, complex, incomplete. Your genes tell the opening chapters, but the rest, your choices, your hopes, your connections, those are still being written. And as science moves forward, we are learning how to read the library with more care, to treat the fragile books with gentleness, to copy what helps, to heal what's broken. That is the wonder of modern genetics. It is not just about data or molecules. It is about meaning, about listening to the whisper inside every cell, and using that knowledge with grace. We end not with an answer, but with a question. What does it mean to know your code? Does it change how you see yourself? Does it change how you see others? Perhaps the more we understand our genes, the more we see ourselves not just as individuals, but as part of something bigger, a long thread winding through time, connecting us to the past, tying us to each other, and pointing gently the future. Let that idea stay with you, softly, quietly, as you drift into rest or rise with renewed curiosity. This is the science of life, and it is just beginning.